be a folder. In that folder will be a data file that I want you to download, and I want you to open up a new R script and be ready to upload that file when the time comes. So we've been talking about two-way analysis of variance, and we kind of finished up last time, which seems like forever ago. We finished up talking about this data set having to do with rat fat. And you'll recall with that data set, let me actually just go and pull it up for you to remind you. This is what the data set looks like. And for each of these combinations of the kind of food that you're feeding these rats and the type of rat that they are, you have 3 replicates for each of them. So we've talked about replication in the past, but I haven't probably used that word at all or very much. But replication, it's the number of times you've observed something. So it's simply the number of times you observe something. But it also sets your sample size. Your operational sample size is based on your replication. So in this case, we have a sample size of 3 because for each of these treatment combinations, we have 3 observations. The total sample size of the whole thing is 12, but those 12 observations are spread across 2 types of fat type and 2 types of gender in this case. And so our operational sample size within sample variation is 3, which is not very much. That's a really low sample size, actually. And so the more you can replicate the observations that you make, the bigger your sample size. That becomes important because when you calculate things like within sample variance, remember it's the sum of the individual values minus the mean squared divided by the sample size minus 1. The bigger your sample size is, the smaller your variance is. The smaller your variance is, the easier it is to tell 2 means apart from one another because they're not being clouded by all of this underlying variation. And so one of the things about designing experiments or designing sampling schemes is you're trying to reduce within sample variation. You're trying to reduce the residual term in your ANOVA table. So when you calculate this ANOVA, you're trying to make this as small as possible. This residual term, remember, is just the sums of all of the within sample variation for all of the different subdivisions of your data. And so that's one of your goals. The degree to which you can do that is the degree to which you'll have the statistical power to detect differences when differences occur. And so that's all well and good because oftentimes we do have projects where we have adequate levels of replication. However, there are some situations where replication just isn't possible. And we also have methods for dealing with that. So in your textbook, there's a whole separate chapter that deals with this, that deals with two-way analysis of variance using what they call randomized block designs. These are situations where you want to do a two-way ANOVA and you don't have any replication. So I'm going to give you a real-life example from some of my own work that illustrates this. So let's go back. So in my past, I've worked in Puerto Rico, and when I've worked in Puerto Rico, I worked with a research group there at the University of Puerto Rico where I was a postdoc, and we worked on hurricane disturbance. So in 1998, approximately a month after I arrived on the island, there was a hurricane that hit the island, Hurricane George. It was devastating. It put my postdoc back. Like I had a 2-year postdoc and it was delayed by 18 months, essentially. But in hurricanes, what happens when a hurricane hits a forest is it opens up the canopy. And in the process of opening up the canopy, two things happen. You basically go from a canopy that looks like this, if you took a fisheye lens photo up into the canopy, and it converts it to a canopy that looks like this. And essentially, all the branches have been broken off of the trees, many trees have fallen down, etc. So when canopy opening occurs, all that canopy that was up in the top of the trees is now down on the ground. And we call this green litter because the day before the hurricane got there, it was alive, it was green like the trees, the leaves on the trees that you see outside. But the other thing that happens when you open up the canopy is that forest that was very shady is now very open. There's no shade from the sun during the day. That leads to microclimatic changes. So you have these two effects as a result of a hurricane. Green litter deposition. This green litter has a lot of nutrients. There's a lot more of it. It's a lot more nutritious. And this can lead to changes in the forest. But then you also have this microclimate issue, which is the canopy is open, so sun reaches the ground more. It gets hotter during the day. It gets cooler at night. It gets wetter when it's wet. It gets drier when it's dry. This is a chart of temperature in treefall gaps where there have been canopy openings. You see it gets much hotter during the day. It gets somewhat cooler at night. In places with closed canopy forest, the temperatures are much more stable. So all of this is going on, and all of these things can affect the organisms that I work on, which are lizards, because if you give a forest more energy and nutrients, it will support more insects, and then the insects are eaten by the lizards. But these lizards are also ectotherms, and so they are in this changed microclimate, and they can't thermoregulate the way they did before. And so both of these things may affect the kind of lizards that I work on. That's the area where we work over there on the far right. And so these are the lizards that we work on. This is a closed canopy species. This is a species that lives up in the very top of the trees where it's nice and bright. This is a species that tends to occur in treefall gaps. It likes highlight, high temperature environments. And this is another treefall gap species. And so there are these different lizards that do different things. And after a hurricane, they may be found in different parts of the forest because the forest has been fundamentally altered. Now, the problem with all of this is that as we study hurricanes, so this is a site that has been studying hurricanes since the early 1980s. As we study hurricanes, a hurricane hits the island, it destroys a bunch of trees, it changes the microclimate, it changes the nutritional value of the litter that's on the forest floor, and we watch these things alter in real time. But what we don't know is we don't know anything about the dynamics of what's going on. Is the dynamics that we see, for example, in fluctuations in lizard populations, is that due to canopy opening and changes in microclimate, or is that due to this translocation of litter down onto the forest floor? So we foolishly got this bright idea that we would try and decouple these two things by doing an experiment. And so this is called the canopy trimming experiment. We have a set of plots that are just control plots. It's got a full forest canopy and it's got a full leaf litter layer at the bottom on the forest floor. Then we have a hurricane simulation where we hired arborists to go in and they cut the canopy out of the forest and then just deposited that onto the landscape. This is basically what happens during a hurricane. You get canopy opening and you get this detritus addition. Then we said, well, how do we decouple these? Well, we go to another section of the forest, we cut the canopy out, but rather than translocating back to the ground, we pick all of that up and we carry it to another set of plots where we haven't gotten rid of the canopy. So this is canopy opening without detrital addition. This is detrital addition without canopy opening. So now this is a two-way analysis of variance. We have two factors, canopy opening or not, and detrital addition or not. And so we should be able to tease these things apart. The only problem is that these plots are big. They're 30 meters by 30 meters. It took us about a year and a half to do all of the cuts and get the whole thing set up. Because they're big plots and because this is a study area where other people are doing things, we could only have a limited number of places to work. We had to work outside of this big gridded area, which is a long-term monitoring plot. So we basically formed three blocks. One is a low elevation block, two higher elevation blocks. These are on ridge lines in the forest. So we have block A, block B, and block C. Within each of these blocks, we have a control. We have a trim only, we have a detritus only, and we have a trim and detritus combination. But that's all within this block. And because this block may be climatically different than this block over here, because it's a couple of ridges over, and because these are high elevation blocks and these are lower elevation blocks, each of these blocks may be different from one another simply because of where they're positioned on the mountain that we work on. And so this is an unreplicated block design because there's only one control plot on this ridge. There's not two or three or four control plots on this ridge. There's one control plot on this ridge. There's one control plot on this ridge. There's one control plot on these two ridges. So this is what it means to have an unreplicated design. It took us a year and a half to set these up and about $600,000. We went with a sample size of 3 because that's what we could afford. And that's also really all the space that we had in the forest for a variety of reasons. We could have put plots over here perhaps, but this was an area that had been clear cut due to logging activities back in the 1930s. Totally different tree community over here than is over here and here. So once again, there are all these constraints, financial constraints, time constraints, history of past land use constraints, such that we just didn't have a lot of space to do all of this. In some cases, in some of these plots on these ridges, the ridges are relatively narrow such that the plot takes up the whole ridge from side to side. And as you get out to the edge of the plot, you're going down this steep embankment down to the stream below. So there are lots of constraints in all of this. So we monitored this thing for 18 months pre-treatment and for 66 months after we did the cuts. So this is one situation where you might find a lack of replication. None of you are molecular biologists, but oftentimes molecular biologists, they do experiments in what we call I forget what they're called. They're plates with little wells in them, and you'll do different replicate things in each of those different wells. But the problem is that you take this plate ultimately, let's say you're doing cell culture, you're culturing cells, you take that plate and you put it in an incubator, and everything on that plate is in a particular spot in the incubator. There may be another 96 well plate someplace else in that incubator, but even within a single incubator, there may be variations in temperature and stuff like that, such that things that are happening to this plate might be different than things that are happening to this plate. Even though this plate and this plate are exactly the same, they may just be different because of where they ended up being put in the incubator. Diligent people doing experiments in incubators, they'll go in and once a day, they will rearrange all the plates so that every plate spends some time in every little environment. But once again, if you're growing cells on a plate, you might actually have a crucial time period during which something happens, and during that time period, all the plates were in a different spot. And so there may be some variation among plates that is due to the plate that you happen to be on. It has nothing to do with what your experiment is. This then becomes a block in the same way that these are blocks. What may be happening here may be different than what's happening here. So by creating a block of observations here, we can isolate that block variation that is due to, not due to any of the treatments that we're applying to those spaces, but just simply due to, in this case, topographical variation. So this is why we need to analyze two-way analysis of variance without replication. And so the calculations for this are pretty straightforward. You have columns and rows. So in the example that we're going to use today, it's a classic non-sciencey sort of randomized block design. We have 4 brands of a product. Let's say pasta. You go to the store, you buy spaghetti. You can get the store brand spaghetti, you can get Barilla, you can get lots of different brands of spaghetti, just your standard size 9 spaghetti. But you can also find these across different stores. So this is the store. These are the brands. And then in each of these cells is the price, whatever the price happens to be, ad nauseum. But of course, at any given moment, this brand in this store, there's only one price. This brand in that same store, there's only one price. This brand in that store, there's only one price. This is by definition unreplicated because all of the spaghetti of that brand in that store is presumably priced the same unless somebody made a mistake with a pricing gun. Of course, we don't even use pricing guns. You guys ever used a pricing gun? Yeah, it's a little gun. It makes little stickers. Anyway, so we might want to know, do different brands of pasta differ? You might want also to know if different stores differ in price for this particular item, pasta. But, of course, one of the advantages of a two-way analysis of variance is that you might want to know if there's an interaction between store and brand. Let's say that brand D is some really high-end Italian pasta imported from Italy that you really, really, really like, and you want to know if there's one store where it's just cheaper so that you can go and get that brand at that store because they have the cheapest price, not on pasta in general, but on that particular brand. That would be an interaction effect. The unfortunate thing is, what do you not see in this ANOVA table? What's not there that's in a normal two-way ANOVA table? Well, F and P values, but this is just formulas. Go back. What do you see in this table that is not in the last table? The interaction term. So there's an interaction term. There's a row by column interaction term here, and in the without replication situation, there is no interaction term. The reason that there is no interaction term is because you don't have replication. If you had lots of observations of different prices of this brand in this store, then you would have degrees of freedom to test the interaction term. But you don't have any degrees of freedom to test the interaction term. So one of the things that you lose in doing a two-way analysis of variance without replication is you lose the ability to test the interaction. So you can test the main effect, in this case, of brands. You can test the main effect of store, but you can't test whether stores and brands interact with one another in ways that maybe there's one store that is particularly cheap for one particular type of brand. All of that interaction variability goes into the residual term. This is relatively straightforward, except in the way in which you calculate the residual term. And just to clarify that for you, I want to run through that equation. The residual term is simply the actual value that is any particular cell. So if we go back, oh, we haven't done it. If this is a particular price, it's this value. Whenever I write Yij, I'm referring to this. It's the value of the ith column in the jth row. And so it's this minus the mean of the row minus the mean of the column plus the grand mean squared, summed up across all of the values, or summed up across all of the rows and summed across all of the columns. And then that all added up is how you get the residual value. All the other things are just the same. We can create row means, we can create column means. We get the sums of squares due to rows by subtracting the row means from the grand mean, squaring that, multiplying or adding all those up, multiplying times the sample size. Once again, column mean minus the grand mean squared times the sample size of the number of rows that you're doing it across. So all of these other ways of calculating sums of squares is the same. It's just the residual sums of squares that is a little different. So when we look at, this is the data file that is in your on the Moodle page. Here we have 4 different brands in 10 different stores. And so we can calculate column means. We can also calculate row means. The sums of squares row is going to be this value minus the grand mean squared plus this value minus the grand mean squared, this value minus the grand mean squared, this value minus the grand mean squared, summed up and multiplied times 4, because you have 4 rows that you're averaging across. The column means is the mean of brand A across all the stores. Sum squares column is just this minus the grand mean squared plus this minus the grand mean squared, this minus the grand mean squared, this minus the grand mean, this minus this, and then multiply times 10 because you have 10 rows that you're doing this across. And so the calculations are not difficult. These are the row means, these are the column means. I'm sorry, I told you that wrong. What I was just telling you, that was, what I just told you was sum squares total. When you take each of these versus the grand mean, that's sum squares total. I had a mental slip. Sum squares total is each of the individual values minus the grand mean squared across all rows and columns. Row means and column means, sums of squares, are actually just sums of squares columns is this minus the grand mean, 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 once again, across all 10 rows And then the row means, the sum of squares rows will be this minus the grand mean, this minus the grand mean, this minus the grand mean, this minus the grand mean across all of those rows. So if we do this, if we take 2.431 minus the grand mean, which where is the grand mean here? The grand mean is 2.459. If we take this minus that, square it, we will get that. Take this minus that, square it, we'll get that. This minus that, square it, we'll get that. This minus that, square it, we'll get that. So these comparisons go into the sum squares total. Now if you add these up, they don't add to 0.05, but then you multiply this times 10 because you have 10 observations that this is being made across. And so that's 10, 18, 18, 18 and 11. 7, 8, 9, 10, 11, 12. 7 is 19. 4, 0, 0. So it's really 0.00499, which we can just round to 0.0005. But then when we multiply this times 10, that's going to give us 0.05. So that's where that 0.05 is coming from. This is just the row mean minus the grand mean squared, row mean minus the grand mean squared, row mean minus the grand mean squared. Each of these things gets all added up. This will not sum to 0.1745, but it will sum to 0.1745 when you multiply it times 4, because you're getting each of these things across four columns of data in the same way that you got this across 10 rows of data. So the sums of squares due to row and column are pretty straightforward to calculate. Sum of the squares total is also really easy to calculate. It's just like you did before. This minus the grand mean squared plus 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 this minus the grand mean squared, this minus the grand mean, 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 plus this minus the grand mean, 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 this. Across the whole data set, the value minus the grand mean squared, all added up, is total sums of squares. Now you could then take sum squares of rows, sum squares due to columns, and sum squares total, and from that generate sum squares residual, just by taking this, and subtracting them from this. You could do it that way, but you could also do this by basically taking 2.43 minus 2.431 minus 2.445 plus 2.459, squaring that. That's the sum square residual for this particular value. It's the value minus the column mean minus the row mean plus the grand mean squared. That's one. Then for the second one, you would do 2.47 minus 2.495 minus 2.445. It's the same because you're still in the same row, plus 2.459 squared. And then just keep adding those together until you've done this for everything in your data set. And when you do that, this is the residual term, remember? the value minus the column minus the row plus the grand mean squared across all rows and columns. And so you will get each of these forms of variation. Sum of squares due to row, sum of squares due to column, sum of squares due to residual, and the total sums of squares, and their respective degrees of freedom. And the degrees of freedom are once again based on the number of columns minus 1, the number of rows minus 1, the number of columns times the number of rows minus 1, and then the total sample size minus 1 is your total degrees of freedom. And so from this whole thing, you would end up getting an ANOVA table that looks like this. In all of these calculations, we have sums of squares due to columns, we have sums of squares due to rows, we have the totals, the residual sums of squares, and we have the total sums of squares. So you should see all of these values appearing in the ANOVA table. We have 4 brands, so we have 3 degrees of freedom for brand. We have 10 supermarkets, so we have 9 degrees of freedom for supermarkets. We have 27 degrees of freedom because 9 times 3 is 27. And we have 40 observations total. So 40 minus 1 is the total degrees of freedom. And you should also see that 27 plus 3 plus 9 equals 9, 10, 11, 12, and 7 is 9, equals 39. So once again, the degrees of freedom within your table should add up to the degrees of freedom that are the total. In the same way that this sum of squares, this sum of squares, and this sum of squares should all add up to this. This is what we calculated for brand, is in this case the column totals. Supermarkets are the row totals, the row means, and then the residual. And then you just do everything like you have done before. You just divide the sum of squares divided by the degrees of freedom. You get mean square, get the F value by taking the mean square divided by residual. Mean square divided by residual. These end up being really, really huge, which gives you a really, really tiny p-value. So in this case, what it's telling you is that brands differ within stores, and brands differ, and prices differ across stores. What it doesn't tell you is how brands and stores differ together in concert with one another. And so you can tell that brand B and C are generally more expensive than brand A and D. So if you know what brands these are, you know which brands are cheaper. It also tells you that some stores are more expensive than other stores are. So there's variation from store to store for this particular product. But what you can't do is you can't measure the interaction. You can't test to see if the rankings of the brands within each store are similar to one another or not as you move from store to store. Now, this is sometimes referred to as an interaction plot, because if these lines are moving in parallel with one another, you can assume that there isn't much of an interaction, even though you can't test that interaction formally, because at this point, each of these points is just the original data. Notice there aren't any error bars around these things. And the reason is because there is no within sample variation, because there's only one observation for each combination of supermarket and brand. But because these things all tend to be moving in parallel with one another, we would assume that there's not a huge interaction going on. Otherwise, you would see these lines crossing one another a lot. And so even though you can get an intuitive feel for whether or not there's an interaction, you can't actually formally test that interaction to see if it's there. All right, questions about that? It's not that different from doing this with replication, except that the within sample variation factor is calculated differently because you don't have any replication. And that's really the only difference. Questions? All right, then. Go to R. Open up a new R script. So open up an R script and read in your data file if you haven't already done that. And since it's a small data file, you can actually just look at it in its entirety. Once again, we have supermarkets, we have brands, we have prices. Put up your red sticky if you're having difficulty getting the data file loaded. Put up your green sticky if it's working. Oh, everybody's got it? Okay. So somebody tell me, how would you specify a two-way analysis of variance to analyze these data? What would be the syntax? I'm going to do two analyses, two different analyses. So I'm going to just name this first one analysis 1. What would the syntax be? Okay, so first thing you need to do is you need to tell it what kind of analysis you're going to do, right? What would that be? Hmm? Yeah, but what is it a linear model? Is it an analysis of variance? What is it? Yep. So we would start off with AOV, and then we would have, what's the response variable? Price, tilde. Okay. Okay. And then we specify where the data are in whatever data frame you saved it in. Okay, let's everybody try that. Here's what I got. Is this what you got? If this is what you got, put your green sticky up. You have to have taken your green stickies down earlier. Green stickies are up. Yeah.
Price not found.
Let me see your data file.
This is
Let me get ahead of it, the top of it. We'll just, or you can scroll up, or do head. Okay, price. Oh, that's weird. Okay, go back to, oh, that's not your data frame. Your data frame is price stat.
I have like some of the right numbers, so it's just
Do summary in front of that.
in front of.
So it's telling you everything you need to know. It's giving you sums of squares and degrees of freedom. But if you do summary in front of it, it'll give you a nice ANOVA style table. If you named your data frame something other than what I named it, then you need to use your data frame. Did it work for you now? Summary. Yeah. All right, who can tell me what is wrong with this ANOVA table? You got it? You got it. Okay, cool. What's jacked up about this ANOVA table? What's it lacking? This is the ANOVA table for this analysis. What is this ANOVA table missing? It's missing F and P values. Why does it not have an F? Fisher statistic. Sir Ronald A. Fisher, the inventor of the ANOVA. Every ANOVA table should produce F statistics. Why is there no F statistic here? What is F? F is the mean squared treatment divided by what? Mean squared residual. So what is your ANOVA table lacking? It's lacking a residual term. So because it's lacking a residual term, it's not going to calculate an F statistic to test the row effect and to test the column effect, because you've taken all of the residual variation and you've stuck it in this interaction term. So you asked for an interaction term, and this is R's way of saying you can't have an interaction term. You're not allowed to have an interaction term. You've used up all of your degrees of freedom, and as a result, if you're going to try and calculate what the effect of the interaction term is, I'm not going to let you do it because you lack the necessary replication. Notice that what is residual in my calculations in the PowerPoint is the exact same thing as what is the brand by supermarket interaction. So Chance, what are we going to change since this was your baby to begin with? What are we going to change about this syntax to get it to give us a residual term? When we first did the two-way analysis of variance, I think it was Blaine who suggested that we specify the analysis as brand plus supermarket. When we specified it that way, it gave us the main effect of brand and it gave us the main effect of supermarket. What did it not give us? I realize that's a long time ago. It didn't give us an interaction term because we used the multiplier as shorthand for give us all the main effects and give us the interactions. So let's just change that to a plus and see what happens. Now this stuff that was in the interaction term is now defined as the residual variation, the variation that is unexplained by brand and unexplained by supermarket. Because that is now a residual, it will do mean squared brand divided by mean squared residual to get that F value. It will do mean squared supermarket divided by mean squared residual to give you that F statistic, and it will test those. So how you specify are doing something matters. And so it's important that you recognize when you have an unreplicated situation versus when you have a replicated situation, because in some cases you will specify it incorrectly and it will give you everything you need to know. You could actually take this table here and say, oh, I screwed that up. That's actually residual, and calculate F and P yourself. But if you expect R to do that for you, you need to make sure that you're giving it the correct specification of the analysis. Brand by supermarket gives you the main effect of brand, gives you the main effect of supermarket, and it gives you the brand by supermarket interaction. Brand plus supermarket just gives you brand and supermarket and nothing else. So in the same way, if you have a two-way ANOVA with replication, you want it to give you the interaction term, so you would use the asterisk. If you have an unreplicated situation, there is no interaction term, so you would use the plus to basically say, R, I don't want you to try and calculate an interaction term. I know that there is no interaction term to be calculated. Okay? So after you finish an analysis, what might you want to do? You know, testing underlying assumptions, especially in a situation like this, because there is no within sample variation, becomes then difficult to test, for example, for normality in your data, because we would normally do that by calculating the residuals for each within cell treatment combination and then sum that up over everything. So you can't do that because there is no within sample variation. So certain assumptions you could test to see if the entire data set had data that were normally distributed. But if there are differences, your normality assumption is probably going to be rejected, not because your data are non-normal, but because you just have treatment effects. You have row effects, you have column effects. And so testing assumptions in unreplicated designs becomes really kind of pointless after a while because you lack within sample variation to test these things. So let's make a graph. How would we start out by making a graph to illustrate these effects? You could, I don't know what that would tell you, but, and what would you be histing? You could just do that price, and it'll give you a histogram, certainly, of all the prices across all the things. But once again, this doesn't look terribly normal But is it not looking terribly normal because the data, the underlying data are inherently non-normal, or is it not looking normal because what you're basically doing is plotting the treatment effects? You're not plotting variation within samples. So once again, this idea of kind of how we think about our data is different in an unreplicated situation. What other kind of graph would we want to make? Yeah, we might want to make a graph of means and confidence intervals, right? So what's our first step in getting us there? Okay, so we'll give it a name. And then what are we going to use to summarize these data? Now you need some data for ggplot to crunch on, right? How have we done this in the past? We want to do summary SE. Summary SE. And what do we have to give summary SE? We have to give it the data file, the data frame, and then what do we have to give it? What's the measure bar in this case? Price. And remember to put it in quotes because whoever did this little routine put their variables in quotes. And then what do we need to do? Group bars. And then what are the group variables? Okay. Run that. Oh, helps if one knows how to spell. Ah, so I get a warning message. It says NaNs produced. So let's look at it. Let me know if you get to this point. Put your green sticky up if you get it to run. When it runs, it gives you this error and it gives you this output. What's the problem?
I probably have something spelled wrong somewhere.
It looks right. Did you run it?
Yeah. I ran and I got an error.
Oh, you don't have RMISC loaded. It's basically saying could not find function, summary SE. When it can't find a function, it's looking for a function that is somewhere in a package.
Okay.
The beginning I had on the board, load RMISC and load ggplot2.
Right.
That's the problem. Yeah, what's going on here?
I got it figured out.
Oh yeah, okay, cool. Awesome. Oh, that's from last time. That work now?
Yeah.
Okay. Oh, okay. So run that now.
Yeah, I still have to load my R.
Oh, you capital
And
R.
then I stuck no R on my head.
Mm-hmm. MISC. MISC. C. Okay, now when you run CI plot, should. Whoa. Library. Brary. BR. You left an R out. Okay, now we go.
Yeah.
Okay, now show it to yourself. Just type in CI plot. Oh, look at all that. Okay, so everybody got this, right? What is the error telling you? No, there are no zeros in your data. It's giving you the supermarket price. It's not giving you standard deviation. It's not giving you standard error. It's not giving you a 95% confidence interval. Why? Those three measures are based on what type of variation? Variation among brands, variation among supermarkets, or variation within the brand supermarket combination? Is it within sample variation that those are based on, or is it among sample variation that those things are based on? What has it produced? In brand A, we have supermarket A, B, C, D, E, F, G, H, I, J. What are the numbers there? 2.43, 2.48, 2.38, 2.4, 2.35, 2.43. What's the sample size for each of these things in your summary SE output? It's a sample size of 1. You can't have within sample variation. What's the equation for within sample variance? It's the sum of the x values minus the mean squared divided by n minus 1. If your sample size is 1, what is this? Well, no, it's 1 minus 1, which is 0. Anything divided by 0 is undefined. Go back to basic math. You can't calculate within sample variation, so you can't calculate a standard deviation if you can't calculate a variance. You can't calculate a standard error if you can't calculate a standard deviation, and you can't calculate a confidence interval if you can't calculate a standard error. This is R's way of, once again, telling you you're asking me to do something that I can't do because I can't do it because you have an unreplicated two-way ANOVA design. So it wouldn't make much sense to basically do this plot, because if we were to do this plot, let's just make a plot real quick. ggplot. We want the data to be in this case, we want the data to be the data from this summary SE output that we just went through. The aesthetics would be x equals, let's say x equals supermarket, y equals price, and we will color these things the color of the brand, and we will plot these as points. And when we get this, it's a yucky looking plot. So let's put some lines. Well, that did not do it. Why is it not giving me? Irritating. Why? Oh, you have to put a grouping variable in order to get it to give you lines. So add a group equals brand to your first statement there. And basically it's giving you this figure that I already showed you in the PowerPoint, which is just basically showing you what the raw data are. It's telling you how brands vary by supermarkets, but there's no variation around those things because there's no variation in those things. For each brand, you only observe that brand once in each supermarket. So there is no within sample variation to plot in this type of way. So homework for Friday, if plotting things this way doesn't make any sense from the standpoint of looking to see if brands are different from one another and looking to see if supermarkets are different from one another, how would you plot these data? Since this isn't giving you kind of a traditional sort of thing. Now you can see that brands do differ. Brands B and C are generally more expensive than brands A and D. You can see that supermarkets differ, but you can't make a plot that shows you the differences among supermarkets, and you don't have a plot that shows you the differences among brands in the way kind of the statistical test would be indicating to you those differences exist. So how would you go about plotting this in a different way? And it may mean that you need to regenerate summary SE output in order to create graphs that are more along the lines of this and more along the lines of this, as opposed to this, which is what we just generated. Okay? All right. We're out of time. I will see you on Friday.